Hello guys, welcome to Agam's webinar on biochemistry session. Today we will be discussing about RNA or ribonucleic acid and a very small topic on mitochondrial genome. So before going to the topic proper, I would like to address certain issues that many of you first year MBBS students might be facing now. Many of you guys are very anxious about your university exams and its postponement or preponement, issues like that. So what I would like to say to you guys is that the university and college is responsible for conducting the exams and it's their headache. So you don't have to worry about the exam conduction or the time of the exam. Just don't waste your time, start preparing now, utilize your time properly and be ready for an exam as soon as this lockdown and everything settle down. And the next issue that many of the students complain is forgetting the subject even after learning. So I would like to tell that MBBS is very vast. You can't master it within a day or a month or even a year. You need consistent efforts and perseverance to master the subjects in medicine. For that there is only one key, that is revision and just revision. You have to keep on revising the concepts until it becomes engraved in your mind and you don't forget it anymore. So these are the two things that I wanted to say, and stop complaining if some topics have not been dealt in your college properly or it has been neglected in your college. In MBBS we don't basically expect a spoon feeding kind of learning. A student-oriented or self-directed learning is what is expected from an MBBS undergraduate. So even if it's not dealt properly in your college, please try to understand that you have to learn it ultimately. You are the person at the end of the day who needs to know the concept. What I want to tell in this regard is like MBBS is just like walking through a tunnel. And we as seniors and your assistant professors, everybody will be just help you to walk through the tunnel. They will caution you about the difficulties, the rocks and stones that you will have to face during your journey. They will help you to get up when you trip down in your journey. But ultimately walking through the tunnel is your journey and it's completely up to you. So everybody, we all are there, your seniors, teachers, everyone is there to support you, to promote you guys and to motivate you people. So stay motivated, stay positive and utilize this time very efficiently for learning your concepts and study the subjects well. So let's come to the topic. Today we want to discuss about RNA or ribonucleic acid. So we know that the most important genetic material for a eukaryotic cell is DNA, but the cell cannot work just with the DNA. RNA is an important factor in the expression of a gene. So first of all, RNA is a single-stranded molecule, and we know that for any genetic material there needs to be a sugar, nucleotide base and phosphate for forming the bond. So the sugar in ribonucleic acid is ribose. On contrary, in DNA we have deoxyribose. Phosphate is same for both DNA and RNA. And if you look at the bases, adenine, guanine and cytosine is same for both DNA and RNA. The only base that is different is instead of thymine, which is present in DNA, RNA have uracil. And all of you know that adenine and guanine are purines and uracil and cytosine are pyrimidines. So this is about the basic structure of RNA or ribonucleic acid. Now one of the most important concepts in molecular biology is the central dogma. DNA undergoes replication to form multiple copies of the DNA, which in turn is transcribed, or transcription process will lead to the formation of RNA, and RNA is converted to protein with the help of translation. And these different types of proteins formed in the body, they affect all the body mechanism, the cellular mechanism, at all the processes at the cellular level. So central dogma is very important. Now coming on to reverse central dogma, this is mainly seen in certain viruses which have RNA as the core genetic material. Here, RNA is converted to DNA with the help of reverse transcriptase enzyme, and from the DNA the protein synthesizing mechanism happens. This is known as reverse central dogma of molecular biology. Now the most important thing that you guys want to know is the latest and modified central dogma of molecular biology. According to this, a genome is converted to transcriptome, which is in turn converted to proteome. So genome includes the entire genetic material present in an organism. Transcriptome includes the entire complement of RNA which is present inside a cell, and protein also refers to the entire collection of and complement of protein which is present in a cell. So in central dogma, the concept was just fixed to RNA, DNA and proteins. But in modified central dogma, we have a very holistic view. We are considering the entire genetic material, the genome, the entire RNA, which is a transcriptome, and the entire complement set of proteins, the proteome. Now let us look into the classification of RNA. So RNA is classified into two different types, basically based on its protein encoding ability. We know that the main function of RNA is protein production. Now, the protein encoding RNA is mainly mRNA. Remaining all types of RNA is non-protein coding. Its function is not directly to code for a protein. It might be either helping in the protein synthesizing mechanism or some other mechanisms. So protein encoding, the one is mRNA, and non-protein coding, we can classify it again based on whether it is large or small. Large we can classify again as rRNA and long non-coding RNA. rRNA and long non-coding RNA. Again, non-protein coding can be classified into small, which in turn consists of the tRNA or the transfer RNA and the miRNA or the microRNA. So here we will be discussing only about mRNA, rRNA and tRNA, which are the most important things that we have to learn for our biochemistry exams and for understanding of genetics. Now let us discuss each RNA in detail. The first one is messenger RNA. From the name itself, it is clear that it carries some message. Okay, so what is the message that is carried? The DNA consists of a gene segment which tells us which has the information for the regulation of the cell. So this information is first converted into RNA, namely a messenger RNA, which is converted into protein that will affect the cellular function. So messenger RNA is the most heterogeneous type of RNA. Why should it be the most heterogeneous? Because there are lots and lots of genes, thousands of genes present in our body. Each will code for a particular and specific mRNA. So the number of mRNA produced will be the most vivid and various types. So it is the most heterogeneous type of RNA, and it occupies 2 to 5% of the total cellular RNA, 2 to 5%. Now Why it is called messenger? Because it conveys the information to the translation machinery, which I just explained now. And finally, the 5' end of it undergoes methyl capping and the 3' end undergoes adenylation. We have learned about the post-translational mechanism of RNA in translation. The post-transcriptional mechanism of RNA in transcription. So, the 5' end will undergo methyl G capping and the 3' end will undergo adenylation by 2 to 250 adenylate residues. So, that is what we have to learn about mRNA. Now, coming to rRNA or ribosomal RNA, it is the most abundant RNA. It constitutes about 80% of the total cellular RNA. It constitutes about 80% of the total cellular RNA. So, what is the function? Ribosomal RNA is important in the formation of ribosome. What is ribosome? Ribosome is a protein manufacturing factory during translation process. Consider that protein is your product, and the company that launches the product protein is your ribosome. And who is the owner of the company? The DNA. Who are the workers? The mRNA. Did you understand the concept? If you take protein as a product released by a company, the company name is ribosome. The owner of the company is DNA, and the workers, the working information of the company is your mRNA. And finally, the product that is going to be released from the company is protein. So, ribosome is formed of RNA and proteins, rRNA specifically, rRNA and proteins. Now, coming to the difference between prokaryotic and eukaryotic RNA, this is another important topic that you have to understand, because it has significant importance when you learn about the drug action in pharmacology. Some of the drugs will be targeting the 30S subunit, some will be targeting the 50S subunit. So, to understand the mechanism of certain drugs, you have to understand about the basic structure of ribosomes. So, prokaryotic ribosome consists of 30S subunit and 50S subunit. Totally it makes 70S type of ribosomes. It consists of 30S subunit and 50S subunits. 30S subunits consist of 16 srRNA and 21 type of proteins. 50S subunits consist of 5 type of sRNA plus 23 sRNA and 31 proteins. So, these are factual things. There is no other go. You have to by heart it. So, what is this S in this subunit? It is known as Swedberg units, which is a coefficient of sedimentation during the centrifugation process. So, going to that much detail of Swedberg unit is not required. We just have to understand the subunit and the protein and RNA present in each subunit. Now, coming on to eukaryotic ribosomes. Eukaryotic ribosome is 80S type of ribosome, and it is formed of 40S subunit and 60S subunit. The 40S subunit consists of 18 srRNA and 30 proteins. 60S subunit consists of 5S RNA, 28 sRNA, 5.8 RNA and 50 type of proteins. So, that's all about the ribosomal RNA, and now we are moving on to the most important topic in this particular chapter, which is the transfer RNA, and this is a part from where you can expect lot of university questions. Transfer RNA is usually asked 4-mark question in the university paper. So, with the new curriculum, it will come as a 5-mark. So, tRNA is also known as adapter RNA. What is the function of transfer RNA? It transfers the amino acid from cytoplasm to the ribosomal protein synthesizing machinery. all the amino acid which is required for protein synthesis is present in your cytoplasm. So it has to be transported into the ribosomal subunit or the factory where the product protein is getting manufactured. So that is done by the transfer RNA. So transfer RNA is basically supplying the raw material to your company. It is a transporting agency. So that is why it is called as transfer RNA. Understand like this. Now coming to the structure of ribosomal RNA, the structure of transfer RNA is very important. It is basically protein. We know that proteins have 4 structural level of organization: primary, secondary, tertiary and quaternary. This is a protein which is present in primary, secondary, tertiary structures only. It doesn't have a quaternary level organization. So the secondary structure, like proteins, the organization happens in transfer RNA. It's not exactly protein. I don't mean to say that it is protein. So in secondary shape, it is clover leaf shaped. So clover leaf almost appears somewhat like this. So the secondary structure appears more like a clover leaf shape, and the tertiary structure is L shaped. So this name of the structure is quite important for your examination. Now the structure of tRNA basically consists of 75 to 95 nucleotides long, 75 to 95 nucleotide long, and the cytoplasm consists of 31 different types of tRNA. Cytoplasm consists of 31 types of tRNA, and mitochondria, which has a separate genome, consists of 21 types of tRNA, 31 and 22. 31 tRNA, and mitochondria consist of 22 types of tRNA molecule. Mitochondria we are going to deal it at the end. Now there are certain abnormal bases or unusual bases which is present in the tRNA. The first one is dihydrouridine, pseudo uridine, inosine and ribothiamidine. Dihydrouridine, pseudo uridine, inosine and ribothiamidine. These are certain unusual bases or the abnormal bases which is present in the tRNA, which is specific to tRNA and is not seen in any other types of RNA. Now coming on to the structure of tRNA, which is very important. As we learned just before, the secondary structure is clover leaf shaped structure, while the tertiary structure is L shaped. Now let's learn the structure in detail. tRNA, first of all, let us begin from the 3' end. We read any molecule like a DNA strand or an RNA strand in particular direction known as 5' to 3' end. So here let's begin from the 3' end. The 3' end of tRNA is known as acceptor arm because it is a site of attachment of an amino acid during the process of transferring amino acid from cytoplasm to the ribosome. That is why it is known as acceptor arm, because it accepts an amino acid and brings it to the ribosomal factory. This site has a nucleotide sequence which is unpaired and it is CCA. There is an unpaired nucleotide sequence CCA which is present on the 3' end. Okay, so we have done the acceptor arm. Now moving on to the next arm, which is the pseudo uridine arm. Pseudo uridine arm is also known as T5C arm. T5C arm. It is required for the binding of aminoacyl tRNA to the ribosome. The activated aminoacyl tRNA should go and bind to the ribosome. So this is with the help of pseudo uridine arm or the T5C arm. Now coming on to the next arm, which is the anticodon arm. we know what is a codon. Codon is a three bases which is present in the DNA. So complementary to the codon, we have anticodon arm which is present on the tRNA. So anticodon arm is a triple nucleotide sequence which is basically complementary to the codon present on the mRNA, or the sequence which is present on the mRNA. So this sequence is read in 3' to 5' direction, whereas an mRNA sequence is read from 5' to 3' direction. That is most important. Now after the anticodon arm, the variable arm or the extra arm, from the name itself it is clear that it is variable in its nucleotide arrangement and sequence. So this variable arm helps to differentiate between different types of tRNA because it is different in the various types of tRNA. Now coming to the last arm, that is the DHU arm or the dihydrouridine arm. So where did I tell dihydrouridine before? I said it is one of the abnormal or the unusual base which is present in the tRNA molecule. So this DHU arm is important for the recognition of amino acid tRNA. DHU is important for the recognition of amino acid tRNA. So we have finished the 5 important parts of tRNA structure: the acceptor arm, pseudo uridine arm or T5C arm, anticodon arm, and finally the recognition site for amino acid tRNA, which is your DHU arm. So let us see an example of anticodon and codon complement. If you look at this image, you can see that the anticodon of the tRNA has a sequence AUG and it is read in the direction 3' to 5' direction, and in mRNA the codon is UAG. So this is complementary. The codon and anticodon have complementary nucleotide bases. So corresponding to the codon, the anticodon will have a particular amino acid which it will bring to the ribosomal protein factory during the process of protein synthesis. So this was all about tRNA. tRNA is a very important question for your university exam. It is a short note, so don't forget to study the topic when you are studying genetics. Now let's come to another important 4 mark of your exam, which is mitochondrial DNA. It is also an important 4 mark. So basically we know that the genetic material is present inside the nucleus. So what is this then? Mitochondrial DNA is extranuclear genetic material. It is a genetic material which is present outside the nucleus, and from the word it is clear that it is present in the mitochondria. Now the genetic material is circular. This is a circular type of DNA. Where else you have studied circular genetic material? Yes, you have read in your bacterial genetics, your prokaryotes, that they have certain circular self-replicating structures called as plasmids. But plasmids were extra-chromosomal. Mitochondrial DNA is extranuclear. Plasmid is present in prokaryote. Mitochondrial DNA is present in prokaryote is present in eukaryotes. Only eukaryotes have well-defined organelles. So it is present in eukaryotes. And plasmid has a significant use in recombinant DNA technology. It can be used as a vector for transferring the gene. But mitochondrial DNA is not used in recombinant DNA technology. These are some basic differences between your mitochondrial DNA and your plasmid. I just said it that so that you understand the concept and you prepare in such a way. Now, this have a different protein synthesizing mechanism from your nuclear genetic material. It has 22 tRNA. Before I said there are 31 tRNA in the cytoplasm, but mitochondria especially have 22 tRNA, and it is an exception to the universal feature of your genetic code. Genetic code universal feature says that a codon will code for only a particular amino acid. So this particular mitochondrial DNA violates the universality of genetic code. For example, if UGA codes for stop codon, it is a terminator codon in the nuclear DNA, but UGA in mitochondrial DNA codes for the amino acid arginine, in mitochondrial DNA. So this is about the violation of universality feature of genetic code by the mitochondrial DNA. Now let us look on to other features of mitochondrial DNA. It exhibits a property called as dichotomy because both mitochondrial and nuclear DNA is important for protein synthesizing mechanism in mitochondria. I'll repeat, mitochondrial DNA as well as the nuclear DNA are important for producing the protein which is present on the mitochondria or important for the cellular reactions in the mitochondria. Mitochondria is a very important organelle concerning our aerobic respiration, which you all have studied, and TCA cycle, everything is related to mitochondria. So this particular feature is called as dichotomy. Dichotomy means two types, both the nuclear and mitochondrial DNA is involved in the protein synthesis mechanism. Now it is important for maternal inheritance. It is an important aspect in maternal inheritance. Why? We know that during fertilization, majority of the cytoplasm is constituted or given by the oocyte, which comes from the maternal side, and majority of the nuclear material is provided by the paternal side. Even though the chromosomes assort independently and they have equal chromosomes from the mother and father, majority of the cytoplasm is contributed by the oocyte, which has a rich cytoplasm, and the nuclear material is contributed by the sperm, which has abundant amount of the genetic material in his head. So maternal inheritance plays a vital role in mitochondrial genomics. This is very important actually if we talk about plants where they inherit lot of maternal features based on this mitochondrial genome, but in humans it doesn't have too much of significance. It is more important in plant genetics. And another striking feature is high rate of mutation in mitochondrial genome or DNA. Now another important feature is heteroplasmy. What is heteroplasmy? The presence of both normal and mutated mitochondrial DNA in different proportion in different cells. I already said mitochondrial DNA has a higher propensity for undergoing mutations. So heteroplasmy tells us that there can be coexistence of normal mitochondrial genome and mutated mitochondrial genome at the same time, but in different proportion and in different cells. So hope the concept is clear. Now coming to an important clinical feature or clinical significance of this mitochondrial genome, the defects in mitochondrial genome can lead to mitochondrial myopathies, a group of disorders called as mitochondrial myopathies. These diseases by itself are not very important, but since it has been given in textbook, I will just mention the names. The Leber's hereditary neuropathy, Leak's syndrome, and myoclonic epilepsy red fiber disease. The disease by itself is not important. You don't have to go into the details of the disease. Just understand that mitochondrial genome mutation can lead to a particular category of diseases called as mitochondrial myopathies, and these diseases are associated with mitochondrial genomic mutations. Then another feature of these mutations is age related. With increase in age, there is higher risk of mutation to the mitochondrial genome, and it is also associated with decreased OXPHOS function. What is OXPHOS? OXPHOS is basically oxidative phosphorylation, which you have learned in detail in your aerobic respiration chapter, and glycolysis, everything you have learned in detail. So it is basically related to age and decreased oxidative phosphorylative function. These are the two risk factors for the mutation in mitochondria. So to conclude, as I said, tRNA and mitochondrial genome are the two university level important questions which you should be well versed with. Other topics, now for the sake of knowledge and understanding the holistic concept of genetics, you will get an overall idea about genetics. And that's all about RNA and mitochondrial genome. Hope everyone have understood the topic. Thank you guys. Thank you so much. Study well, become great clinicians. All the best wishes. Do well, excel in your life and career. Thank you.